problem. Thank you.
So,
hello, everybody.
Yeah, Dr. Vaccari.
Thanks.
Go
so
ahead.
today I'll be talking about how we can target different pattern recognition receptors to improve outcomes or decrease inflammation after CNS injury and disease. First, I have to disclose my conflict of interest. I'm a co-founder for Inflammacor, which is a company, a spin-off company from the University of Miami that we developed to come up with therapies to treat inflammation after CNS injury, targeting the innate immune response. So I'll be talking about node-like receptors, Rig-like receptors, and C-type lectin receptors in the context of CNS injury and disease. We all are familiar with the innate and adaptive immune responses. I'll be focusing on pattern recognition receptors, such as Tog-like receptors, node-like receptors, or Rig-like receptors. Tog-like receptors are the mostly studied pattern recognition receptors. And upon activation of these pattern recognition receptors, you have cytokines and interferons that then are secreted and then go on to activate an adaptive immune response. These pattern recognition receptors could be in the cell membrane, such as Toll-like receptors here, or even in endosomal membranes, like Toll-like receptor 3. There are also cytoplasmic receptors, like the Node 1 and Node 2, and the NALPs, or the inflammasomes, that are also cytoplasmic, as well as the RLR receptors, RIG1 and MDA5. They are activated by either PAMPs or DAMPs. PAMPs stand for pathogen-associated molecular patterns, and DAMPs stands for danger-associated molecular patterns. PAMPs, examples of PAMPs include LPS, or moramidate peptide, and DAMPs, for example, it could include heat shock proteins or ATP, anything that is endogenous to the system. So I'll start talking about NLR signaling, or NLRs, which are famous for making inflammasomes or being compressing elements of the inflammasome. Back when I started studying the inflammasome back in 2005, there were only about 9 papers on the inflammasome. Nowadays, there's over 3,000 papers on the inflammasome. And it's a multiprotein complex that in humans is comprised of NALP1 or NLRP1, ASC, caspase-5, and caspase-1. This multiprotein complex comes together activating caspase-1, resulting in the cleavage or processing of the pro-inflammatory cytokines interleukin-1 beta and interleukin-18. For instance, interleukin-1 beta, we're familiar with it, and it has a wide array of physiological or pathological consequences. For instance, it stimulates endothelial cells to release interleukin-6, which then goes on to increase the production of acute phase proteins from the liver that is seen as an increased inflammatory response. Also stimulates neutrophilia, or even in the hypothalamic thermoregulatory center, IL-1 beta is responsible for the activation of fever. So we've shown that spinal cord injury induces activation and processing of the inflammasome. To look at inflammasome activation, we look at cleavage of caspase-1. This is a time sequence where we use sham unoperated animals, and then animals that were injured and sacrificed at different time points after spinal cord injury. As you can see, there is an increase in the elevation of inflammasome activation all the way up to 3 days. And inflammasomes are present in different cell types, microglia, astrocytes, oligodendrocytes. And initially, we started focusing on inflammasomes being active in neurons after spinal cord injury. Here you have sham animals for caspase-1, caspase-11, ASC and NALP1, nowadays known as NLRP1. And 6 hours after spinal cord injury, you can see there is a redistribution of these proteins in motor neurons of the spinal cord towards the cell periphery, probably associated with the role that these proteins play on secretion of interleukin-1 beta. And these proteins themselves, nowadays, we know that they are also secreted as well. So we can target the inflammasome by using, for instance, a neutralizing antibody against the adaptor protein ASC. And when we did that, here we have 3 groups. We have animals that were not treated, animals that were treated with an antibody, and animals that were treated with immunoglobulin as a control. And you can see that the levels of caspase-1, IL-1 beta, IL-18, and cleave-XIAP were lowered in the anti-ASC treated group. We quantify this with a fluorometric caspase-1 activity assay. So we can target inflammation with neutralizing antibodies to inhibit inflammation, which results in improved histopathological outcomes. Here you have the motor neuron area, or the ventral horn, which has the motor neurons. And you can see that in the anti-ASC animals, the histopathology is much better compared to the non-treated group. And this is a naive group for comparison. Again, serological analysis was done, showing that less damage took place when in the treated anti-ASC treated group. So we've gone on to show that we can target inflammasomes to improve, to decrease inflammation and improve histopathology and improve function after spinal cord injury, after stroke, as well as after traumatic brain injury. Another pattern recognition receptor that we've looked at are the RIG-like receptors, which include RIG1 and MDA5, which go on to activate type 1 interferons like interferon alpha, interferon beta, as well as interferon-stimulated genes, which are associated with apoptosis or chemokine production, as well as the complement cascade. So targeting this these receptors, we can also target a variety of cytokines. Again, after spinal cord injury, we've seen that these cytokines are present in astrocytes, in neurons, as well as astrocytes. Here you can see the injured group on the right side for the pattern recognition receptors RIG1 and MDA5, as well as the adapter protein VISA, also known as IPS1 or MAPS. We can stimulate astrocytes in culture with a synthetic ligand that mimics double-strand RNA, such as poly-IC, of either low molecular weight, which activates RIG1, or high molecular weight, which activates MDA5. And the stimulation of these receptors results in increased expression of GFAP and vimentin associated with changing morphology of the astrocytes. So stimulation of this pathway affects astrocyte activation. We've also you can, it turns out that after middle cerebral artery occlusion, RIG1 is elevated, associated with an increase in interferon alpha production following this stroke model. And also we can do the same thing in culture in an oxygen glucose deprivation experiment where we grow astrocytes and challenge them with oxygen glucose deprivation for different lengths of time, and that it resulted in increased RIG1 as well as consistent with increased expression of interferon alpha, probably through an NF-kappa B dependent manner. And interestingly, when we looked at samples from plasma samples and temporal cortex samples from patients or descendants with Alzheimer's disease, we had different groups. We had a non-cognitive impairment, mild cognitive impairment, as well as the Alzheimer's disease group. And you can see that there is, when you look at the MCI group, there was an increased expression of RIG1 for the plasma and the temporal cortex. Whereas if you are not to look at that mild cognitive impairment group, you would just say that Alzheimer's does not, that this particular RIG1 protein does not play a role in Alzheimer's. So it's important to look at the early phases of cognitive decline for these studies. So it might be interesting to explore what would happen if we inhibit RIG1 signaling in the mild cognitive impaired patients. One inhibitor that can be tried is propyl gallate, which has antioxidative properties. We tested it after oxygen glucose deprivation, and it results in decreased expression of interferon alpha, for instance. And then finally, C-like receptor, C-type lectin receptors. The one example includes dectin, and the one that I've been focusing on is Minkel signaling, also referred to as CLEC4E. And it's been shown that it's activated by SAP130, which is a transcription factor that, after cell damage, is released and binds to Minkel, and through phosphorylation of SIG, results in production of tumor necrosis factor. And looking at samples from either brain tissue or cerebrospinal fluid from patients with traumatic brain injury, we've seen that there is an increase in the expression, at least in the brain, of Minkel and phospho-SIG, as well as also increase of Minkel and SAP130 in the cerebrospinal fluids. So these proteins definitely play a role also in traumatic brain injury. Using the fluid percussion brain injury, we looked at the expression of these key Minkel signaling proteins, such as SAP130, Minkel, and phospho-SIG, And as we know, TNF is already elevated after traumatic brain injury. So you can see that the expression of these proteins is elevated as a result of traumatic brain injury, consistent with our results in patients with TBI. So since TNF is such an important target, anti-inflammatory target that we could focus on, then we used MiNK antibody to neutralize the effects of SAB130-mediated activation of MiNK signaling. So we had an unstimulated group, the IgG group as a control, and the antibody-treated group at a couple of concentrations in the presence of SAB130 to activate the pathway. And as you can see, it was a decrease in phospho-Syk expression, as well as decreased expression of the TNF group in the anti-MiNK treated cultures. These were neuronal cultures. So today I've shown you that we can target the innate immune response either at the level of pattern recognition receptors to inhibit production of cytokines, such as interleukin-1 beta, interleukin-18, type 1 interferons like interferon alpha, interferon beta, or also TNF. So this is encouraging, and as a result, we're looking at ways in which we can develop therapies to improve outcomes after CNS injury and disease, such as brain injury, spinal cord injury, stroke, or Alzheimer's disease. This is a result of a big effort with a lot of people that were involved throughout the years in these projects, especially the team at the Miami Project to Cure Paralysis in the Department of Neurosurgery and the Department of Physiology and Biophysics, as well as collaborations with Steve Lacroix in the Department of Molecular Medicine at Laval University. Thank you very much.